Hello everyone, and welcome to this podcast for cell biology. In this podcast we're going to talk about or continue our discussion of cellular membranes. And two broad topics that we're going to talk about are first, membrane proteins, the different kinds of membrane proteins, the function of membrane proteins, how we study some of these membrane proteins, and then we'll continue a discussion we started with the last set of podcasts, and that topic is membrane transport. Last time we focused only on diffusion and osmosis. Now we're going to focus on membrane transport that requires membrane proteins. Okay, so let's go ahead and begin our discussion of membrane protein. Before we jump into membrane proteins, let's talk about these two models about the structure of the membrane. The first was the sandwich model, and the other is the fluid mosaic model. I've already mentioned the fluid mosaic model in the past, but now let's talk about what these two models are and why the evidence suggests that the fluid mosaic model is the most accurate way to describe the membrane and membrane proteins. Let's draw a simple membrane here for the sandwich model. Remember, we'll still have these fatty acids with the hydrophilic heads that I just drew, and now the hydrophobic tails. And actually the fluid mosaic model also has that as well. And I'm going to leave a few gaps here intentionally, and you'll see why in a moment. But still the polar heads, the hydrophilic heads here, and then the hydrophobic tails. Now the sandwich model suggests that the outside of the membrane, as well as the inside of the membrane, if this were the inside of the cell, so I can write cytosol here, are lined with hydrophilic proteins that will interact with the cytosol on the inside or with the what we call the extracellular space on the outside. So both hydrophilic proteins. In a way to sort of protect the hydrophobic nature of these lipids. This model was proposed in 1935, and it wasn't until 1972 that a new model, the fluid mosaic model, gathered people's attention. And the fluid mosaic model suggested that certainly there are proteins that are on the outside like so, but there might also be proteins that span the membrane, something like this. And you might have some proteins on the inside. You might also have some parts of the membrane with no proteins. The sandwich model would suggest that proteins line both the inside and the outside of a cell membrane. We also might have some proteins that just partially insert into the membrane. And on the other side are just lipids. Fluid, because these proteins would move along the membrane. We already talked about the fluid nature of the membrane. Mosaic, because mosaic essentially means that something is made of different components and there's not an equal distribution of them. If you look up on the internet mosaic art, it's going to be art that's composed of different materials, different sizes, different shapes, and that's what this model proposed that the membrane was. Okay, now in an attempt to distinguish which of these two models was correct, they did an experiment called a FreezeFracture Experiment. Essentially what they do here is they freeze the cell. So we have our cell here, and it's frozen. They can now cut this cell in very thin sections. More thin than what I'm drawing here. So they get very fine sections of a cell, very, very thin sections of that cell. And what happens if you cut this cell in various places, rather if this sandwich model was correct and you cut the cell, what you would see is a very evenly distributed membrane. Particularly when you ripped out the middle here and you looked at it under an electron microscope. Let me write that word down here. The electron microscope lets you see the cells in a very fine detail, what you would see is this nice even distribution of membranes. And on the EM, even though you see it with pretty fine detail, you're not going to see the individual tails. You're more likely to see something like this, a very smooth structure. Now, after they did the experiments, they didn't see this. They saw something that was more reminiscent of the fluid mosaic model. They froze the cell, they then cut slices through here and looked at individual pieces of membrane. So let's say on one of these electron microscope images, they got the top part of this membrane, the first half of this lipid bilayer. And let's say in another section, down here somewhere, they got this second half of the lipid bilayer. And again, remember, you're just looking at a very thin section that encompasses, very small part of that membrane. And so what they saw was something like this, where on the top, what they would have seen is the lipid membrane and two things that they would have called mounds. They weren't exactly sure at the time what they were, but we now know that they are proteins. Because as you're looking at this section through here, you're going to include some of this protein here, which is going to stick up, and it will appear much different than the membrane around it. Same thing here, you're picking up this membrane and this membrane. This protein and this protein. Now when you split this membrane in half because you're cutting through it, you're also going to be able to see this bottom half of the membrane. And this bottom half of the membrane, you may pick up something in this size, this thickness, all the way across. And on the electron micrograph, what they're going to see is something like this. They'll probably still pick up this protein because it's a transmembrane protein. It goes through the membrane, and we'll define what a transmembrane protein is later. But it goes through the membrane, so you'll still pick up that one. But what happens on this side here, as they split this membrane in half, this pulled over to this first section, not this one. So kind of plucked away, and it left this big gap here. And that big gap here, I'm just going to leave a space there to represent that, is something the researchers called a pit. Because it appeared kind of as a pit, something that kind of sunk in. And the reason that it did that is because at one time, this protein extended into this area. But when you split the cells, cut the cell in half, this whole protein got pulled away with the other half of the membrane, leaving this half of the membrane void of that protein. So the take-home point from this is that there were two models, the sandwich and the fluid mosaic model. When they froze the cells and sliced the cells and looked at them under a microscope, what they found was evidence that the distribution of proteins was not even, as you would predict with the sandwich model, but rather there was evidence that proteins were going through the membrane, or partially going through the membrane, as shown by these mounds and pits in the electron microscope images. Okay, now that we've introduced these proteins and talked about some evidence that suggests that they go through the membrane or partially go through the membrane or sometimes just sit on the outside of the membrane, let's address some of the functions of membrane proteins. Let's talk about the function of these proteins. Just like the lipids had many different functions, these proteins also have lots of functions. They can serve as receptors or signaling proteins. So let me draw a cell down here, and we could draw the signaling protein like so. And there might be some signal that comes here, interacts with the outside of the cell, and transmits that signal to the inside. There's got to be a way to communicate the outside world of the cell to the inside world of the cell. And these receptor proteins are one of these strategies. The next one we should talk about are structural proteins. And I'll give an example here of spectrin. And what spectrin does is it lines the outside, or rather the inside, of the plasma membrane. It's a cytoskeletal protein that helps provide structure and integrity to the cell. The third function I want to talk about is that of proteins acting as a transporter. A good example of this are the glucose transporters, where glucose, which cannot normally go across the membrane, uses these transport proteins to move glucose into the cell. Next, I want to bring up the example of enzymes. We've of course have talked about enzymes a lot, and we will continue to talk about enzymes a lot, but enzymes can sit right here on the border of the plasma membrane and carry out enzymatic reactions. A good example of this is adenyl cyclase. Adenyl will catalyze the reaction to make cellular CAMP, cyclic AMP. This is a very important signaling molecule in the cell. Let's see, we have a couple more I want to talk about. We have proteins that we're going to put in this electron transport category. These could be proteins in the mitochondria, the chloroplast, or bacteria. Bacteria, they don't have organelles, but these electron transport proteins would be found on the plasma membrane. But let's go ahead and draw a lovely mitochondria here, and these transport proteins will be found on this inner membrane. Another category, I'm going to lump together adhesion and communication. These are going to be found on this cell's edge here, and what they will do is they will allow for the adhesion to a neighboring cell, like so they stick together, and those would be the roles of some tight junctions. However, there are other ones that allow for communication, to allow small ions to move from cell to cell, and those would be gap junctions, and I'm just going to put GJ there for gap junction. Now there are of course other functions that we could include here, but I'm going to go ahead and stop here with our list of functions. Okay, now that we've talked about the different functions of membrane proteins, let's talk about the different classes of membrane proteins. And to do that, let's draw our good friend, the famous membrane. Okay, so here's our membrane, but now let's talk about the three classes of membrane proteins. The first one I want to talk about are membranes called transmembrane proteins. I'm going to lump in integral proteins in this category as well. Transmembrane proteins are proteins that can span the membrane. And we'll talk about the unique features of a protein that spans a membrane in a little bit. But essentially what we see here is a membrane protein that goes from one side of the cell to the other side of the cell. Sometimes a membrane protein might only partially go through the membrane. The next class of proteins I want to talk about are the peripheral membrane proteins. These peripheral membrane proteins might interact with a transmembrane protein. And these interactions here are going to be those non-covalent bonds. Van der Waals, hydrogen bonds, ionic bonds that are going to hold these together. So why don't I write that underneath here? Non-covalent. And I didn't say it up here, but it's never too late to go back and edit. The transmembrane proteins are largely positioned due to hydrophobic effects. We'll come back to this later, but essentially what I'm saying is that the amino acids of these proteins, these transmembrane proteins, the part that is in the membrane are hydrophobic, or you could call them nonpolar amino acids. Those that try to get away from the water, or rather exclude water from their association. You could also have these peripheral proteins loosely associated with the fatty acids as well. Now this last class of membrane proteins I'm going to talk about are called lipid-linked. These lipid-linked proteins are also on the outside of the cell, but what makes them different than the peripheral membrane protein is that they are covalently linked to the fatty acids. So they're held together by covalent bonds. Okay, let's say that you are a protein biologist and you're trying to figure out if the protein you're studying is a transmembrane protein, a peripheral membrane protein, or a lipid-linked protein. What you might do is apply certain chemicals to your cells to determine if the cells will be released, or rather if the protein will be released from the membrane. So let's start with a detergent. When you apply a detergent to a cell, it essentially busts the membrane apart. So detergents will release a transmembrane protein. It will also release a peripheral membrane protein and a lipid-linked protein. So how do we know what we're looking at, since detergents release all of those? Well, we could add a salt next. Salts help break up weak non-covalent interactions, but they're not going to break apart covalent bonds or hydrophobic interactions. So when we add a salt to the cells, it won't release our transmembrane proteins, and it won't release our lipid-linked proteins, but it will release our peripheral membrane proteins because they're being held together by these non-covalent bonds. Well, still though, how are you going to distinguish between a transmembrane protein and a lipid-linked protein? There are specific enzymes that will cut specifically at the covalent bonds that hold lipid-linked proteins to a membrane. In this case here, the transmembrane protein won't be released. The peripheral membrane protein won't be released with just an enzyme added, but the lipid-linked ones will. If you're studying a protein and salts don't release them, and the lipid enzymes don't release them, but the detergents do, then you know you're looking at transmembrane protein. Salt releases them, you know it's a peripheral membrane protein. If the lipid enzymes release them, then you know it is a lipid-linked protein. All right, I'd like to spend a little bit more time talking about transmembrane proteins before we move on to membrane transport. I'm going to draw our membrane, but I'm not going to draw all the details to it that we usually draw. I'm just going to draw the polar heads here. And you know that the fatty acid tails are in the middle. But I do want to remind you that in the middle here is nonpolar, and these polar heads on the top are polar. Now this membrane, transmembrane protein that goes from one end of the membrane to the other end of the membrane, often doesn't have to. We could have other structures here, but a common motif here is that it forms these alpha helices, the part that spans the membrane. The alpha helix, remember, forms due to hydrogen bonds along the protein backbone. The amino acids can be polar or nonpolar amino acids. But if it's going to span the membrane, these amino acids have to be nonpolar. Or sometimes we also call those hydrophobic amino acids. They have to be nonpolar so they can solubilize within the nonpolar environment of the membrane. Up here, they can be polar or nonpolar depending on what they're interacting with. But the part that spans the membrane has to be nonpolar. And also important is that typically, for most membranes, the number of nonpolar amino acids that is long enough to span the membrane is 20 amino acids. It can be more, but at a minimum, it has to be 20 amino acids. Now, because of this nature of these amino acids, we would call these proteins amphipathic, meaning nonpolar here in this part of it, and then we could have polar amino acids on either side here. And you certainly would have some polar amino acids here to interact with water molecules. Now, when we talk about membrane transport, we're going to talk about channel proteins. And we'll describe those in just a moment. But essentially, a channel protein is a protein that forms a channel between the two halves of the membrane. And so you might have, say, one alpha helix, see here, and one alpha helix here. Now remember, as I just said, the amino acids on an alpha helix can be nonpolar or polar. And we will have amino acids extending here in this direction as well as this direction. So this just complicates it a little bit from what we saw over here. The amino acids on this side of the alpha helix and on this side of the alpha helix have to solubilize in the nonpolar region of these fatty acid tails. So the amino acids on this side, they must be nonpolar, as well as over here. The amino acids that are stuck in this channel here, the channel that's connecting the outside of the cell with the inside of the cell, these are going to be polar amino acids. This allows water molecules or other polar molecules to freely move down and across this channel in either this direction or this direction. But the alpha helices that form channels have to have polar and nonpolar amino acids associated with them so that they can do two important traits. The first, to solubilize with the fatty acid tails, and next to allow for the movement of polar molecules across the membrane.